And we're going to spend the next couple of minutes talking about some things from peptide and protein folding. This is a topic that you will cover briefly in biochemistry, but it's sort of a graduate level topic. Nevertheless, it's the focus of my research, and I can't help myself. Now, I will say something about these next. We've changed gears, and if you haven't sensed it yet, you will. We've started talking about biomolecules. And the major classes of biomolecules are proteins, carbohydrates, lipids, and nucleic acids. And there's a chapter on each of them. Each of them could be a textbook, and so we're necessarily skimming the surface. And we've almost fallen back to the level of facts and descriptors rather than reactions. That's okay. We need to understand the molecules first before we can do reactions. We will do a chapter on metabolism where we will see reactions mostly having to do with carbohydrates and lipids. But in these chapters on biomolecules, you're going to sort of wonder, what do I have to learn? Because anytime someone presents facts to you, you can get stressed out thinking, am I responsible for memorizing that fact? Some of it, absolutely. I already told you, memorize the 20 amino acids. You don't want to? I don't care. Memorize the side chain pKa's. You don't want to? I don't care. Some of you, by the way, want to know, to what decimal place do I need to know the side chain pKa's? And I respond, are you kidding me? No decimal places. One significant, well, the ones place is sufficient, please. So there are facts that are important, but always more important than those facts will be the use of those facts to draw scientific conclusions. We're going to talk today about the relationship between protein sequence, structure, and function. Try to find the overarching principles that are based on things you already know. Try not to get sidetracked with, am I responsible for memorizing that this particular amino acid sequence does whatever. I mean, if you want me to say yes, I can, but the answer is no. But you're supposed to understand the basic principles and be able to use those basic principles to come up with hypotheses and draw conclusions. So, I present to you a 33 residue amino acid sequence. Have you memorized it yet? I'm serious. I'm not serious. Really not serious. Each of these amino acids is represented by a one-letter code. This is a real amino acid, real peptide derived from the yeast transcription factor called GCN4. It's from the leucine zipper domain of that yeast transcription factor. You may learn about it in biochemistry. If you look at the sequence and understand the categories of amino acids we talked about last time, including those that are nonpolar and hydrophobic, a pattern emerges. I have highlighted in orange a repeating pattern of every 7 amino acids. We see a nonpolar amino acid side chain. Methionine, valine, valine, valine. This one in the middle breaks the pattern, but that's the exception that proves the rule, and we may have something to say about that later. I may just skip over it. And then slightly offset from the orange nonpolar residues, you have another repeating pattern of nonpolar amino acids. Amino acids, sometimes people will use the term residue. That's the section of the amino acid that's in the peptide. It's no longer an amino acid, it's in an amide bond. Don't be distracted by that phrase residue. It just means a specific amino acid within the context of a protein. But I have a leucine here, and then 7 residues away, spaces away, I have this leucine, then another 7, I have this one, then another 7, I have this one. So there's this repeating pattern of every 7 residues, you've got a nonpolar amino acid, and you've got 2 such patterns interdigitated with each other. This is called a heptad repeat, heptad from the Greek prefix meaning 7. And within this repeating sequence pattern, the first and then the 4th amino acids are nonpolar. So methionine at a so-called A position, B, C, D, there's another nonpolar, E, F, G, then nonpolar A, B, C, D, nonpolar, and so on. This is called the heptad repeat. Now, just imagine what having nonpolar amino acids might do to a sequence like this. You know from chapter 3 in 351, you've learned about the hydrophobic effect, how nonpolar surface area prefers not to be exposed to water because exposed nonpolar surface area in water causes water molecules to order themselves around the nonpolar surface, losing entropy. And so to prevent that, the nonpolar stuff will sequester itself away from water. We've learned about micelles. We'll talk about bilayers later. In the same way, you might imagine that this peptide might be driven to alter its shape in such a way that the nonpolar amino acids could be sequestered from water. And this is indeed what happens, and burying nonpolar surface area from water is a key principle in protein folding. It is a key determining factor for why proteins adopt the specific shapes that they do. So let's think about how this sequence might alter its shape to protect these nonpolar pink and orange side chains from water. Remember the pattern though, the space between the A position nonpolar residue and the D position nonpolar residue is 3 spaces, and then the spacing between the D and the next A is 4. So 3 and then 4, 3 and then 4, that's important. Now, your text says some things about peptide and protein secondary structure. We're going to talk about just the alpha helix today. You should be familiar with the beta sheet. We just don't have time to talk about it in detail. So here is a close-up image of just the backbone of a peptide in an alpha helical conformation. And it looks like I have lots of notes written on it from a previous class. We'll erase it to make it clearer. The alpha helix is characterized by this spiral backbone of the peptide. So to get you oriented, this is the amide nitrogen of an amino acid. This is the alpha carbon. This is the carbonyl carbon. This is the oxygen. And we will call that we're going to count from here. This is our starting point. We're going to call this the I position. That's position I, the starting point. Okay, so that would make, and at this point, I'm just going to denote each residue of just with the alpha carbon, because that's easiest to point out. This is the I+1 alpha carbon. And behind here in pink is the I+2 alpha carbon. Then here in green is the I+3 alpha carbon. And then I'm running out of colors. Oh, purple. Here in purple is the I+4 alpha carbon. Notice that spiral pattern, and notice that by the time I get to the I+3 and I+4 position, I'm hovering over that original I position. So in the alpha helix, one turn of the helix brings you back around to sort of where you were to begin with, only now you're offset upwards. That's what a spiral means, right? And this I+4 and I+3 position sort of hover over and straddle the I position. All right. Notice that the carbonyl oxygen, this image is obtained from a crystal structure of this peptide. Notice that the carbonyl oxygen of the I position amino acid is pointed towards the amide nitrogen of the I+4 amino acid. In crystal structures, you don't actually see hydrogens. They're too small. So you have to infer where they are based on bond angles and where the other atoms are. But there's a hydrogen here. And if you measure the distance between this nitrogen and this oxygen, it's consistent with a hydrogen bond between the I position CO carbonyl oxygen and then the I+4 position amide nitrogen. So that hydrogen bonding pattern is characteristic of the alpha helix, And it's not just that one. Look at all of the carbonyls in the alpha helix. They're all pointed upwards towards the amide nitrogen that is 4 spaces ahead in the sequence. Yeah?
So above on the sequence that you gave us, every 7 amino acids were nonpolar.
Yeah.
And we're displaying the hydrophobic effect. It wouldn't be so that's primary structure, and then the
Right.
alpha helix is a secondary structure. But and on that alpha helix, having it every 7 would probably have all of those nonpolars on the same side.
Good.
And so, it wouldn't be revealed until a tertiary structure. That's why it is every 7 steps, or
Yes, that's excellent. The sequence of the amino acids is called primary structure or primary sequence. This is secondary structure. We'll talk about tertiary structure, which is where elements of secondary structure come together. But one point you're making is where I was going, which is if every 3 or 4 amino acids brings you back to the same side of the helix, that means this sequence should put the M and the L and the V basically in a line, maybe a bit of a zigzag along one face of the alpha helix. So that means if this sequence, which has this spacing of nonpolar amino acids, folds into an alpha helix, one side will have a stripe or a zigzag of nonpolar amino acids. And that's key. I wasn't quite done talking about the alpha helix. You can see that each carbonyl is hydrogen bonded to an amide proton that's 4 residues away. This is called I to I plus 4 hydrogen bonding. It's characteristic of an alpha helix. I won't say, but this is a whole area. Each amino acid within this alpha helix has a characteristic set of phi and psi dihedral angles. You can actually identify alpha helices by measuring those dihedral angles. But if the I plus 3 and the I plus 4 residues sort of straddle in space the position of the I residue, that means there's about 3.5 amino acids per turn in this helix, or about 7 amino acids per 2 turns. Remember our sequence pattern of 7 residues. That means if this sequence were to fold into an alpha helix, this first heptad would comprise 2 turns of the helix, and then the next would comprise 2 turns of the helix. And every 2 turns of the helix, you come back in space to an A position or to where the first nonpolar amino acid was. So you can actually, if we look at the helix from another perspective, looking down the axis of the helix, we can see this placement of the A and D position residues all along the same face of the helix more clearly. So that's illustrated here. We start with the A position methionine. The helix goes around to this B position lysine, C position glutamine, and then a D position leucine. Notice we've already made 1 turn of the helix, so the helix is spiraling around away from you into the page. We're looking at the helix from its N terminus. This is methionine 2, this is lysine 3, glutamine 4, leucine 5, glutamate 6, aspartate 7, and then if we were to continue on, we would see another valine, and it would be right back here in the sort of same area where the first methionine was, only 2 turns of the helix further on. This sort of view of an alpha helix is called a helical wheel diagram, and you can turn it into a prettier picture and just list each of the A position residues starting closest to the N terminus and moving outward towards the C terminus, and then starting at the N terminus and moving outward towards the C terminus. And you can see that all of those orange and pink residues are aligned along the same face of the helix. We have a helix that is what my PhD advisor used to call globally amphipathic. Globally meaning overall, amphipathic meaning two characters or two loves, a hydrophobic face, which is this one. And then if you look at all of the other positions, E, F, G, and B and C, they're all mostly polar. You should get to the point where you see these Ks and you're like positive charge, or you see the E's and you think negative charge, and you can see that those are polar. So the other residues here are highly charged, highly polar. So you sort of have a hydrophilic surface here. Now, what does that get you? You've now got a helix that's got a hydrophobic surface on one side and a hydrophilic surface on the other. So far, everything's still exposed to water. Not so if you can bring two such helices together and have their hydrophobic faces interact with each other but be sequestered from solvent. This is like a slide from my PhD defense from like 15 years ago. No, more than that, but anyway. So I present to you your first quaternary structure. We skipped tertiary. Why did we skip tertiary? Quaternary structure means two different molecules engaging in an intermolecular interaction. So one alpha helix binding to another alpha helix. Turns out two of these GCN4 peptides will find each other, and they will have their nonpolar surfaces interact with each other, burying them from water, excluding water, and creating what's called a coiled coil. Why is it two coils? Well, you have two individual coils or helices that are actually wrapped around each other. Here is what that looks like. The interior that I've shown here are all of these A and D positions. Here's that methionine amino acid, and then here's the next leucine. Notice that the leucine, it's leucine 5 on one helix, is packing up against leucine 5 on the next helix. If we had time, I'd show you this in a program called PyMol. We would zoom in, we'd look at it from different perspectives, but we don't. Notice that all of the other hydrophilic positions are all exposed to water. So this folding is driven by the hydrophobic effect. It turns out that any peptide that has this pattern of nonpolar residues might form a coil-coil. The hydrophobic effect provides the driving force for this thing to fold into a helix in the first place. Okay, so you should understand the hydrophobic effect. You should understand, given these facts about helical secondary structure, you should be able to work out how all of the nonpolar residues would be basically along the same face of the helix, and it should make sense to you that two such helices packing against each other would necessarily have a hydrophobic core where the nonpolar residues were protected. There's a ton more to say. We don't really have a huge amount of time for it. I will say that there are lots of different coils in nature, and many of them differ in the identity of the amino acids at the core. In the early 90s, it was discovered that very modest sequence changes for the amino acids at positions A and D in the core of a coil-coil could cause the coil-coil to form not just a dimer, but also a trimer or a tetramer or some more elaborate, larger structures. I'd love to tell you more about that. I just want to illustrate how subtle it is. I will tell you one thing which you don't need to memorize about a modest sequence change that allows this dimer to become this tetramer. And I won't, as I said, I won't say too much about it. Going back to the dimer structure, I want you to notice that at the A position, we have a lot of valines. And valines are an example of an amino acid that we talked about that has, there's the alpha carbon, there's the beta carbon. We call these beta branched. Whereas the leucines, if you've memorized the leucine side chain structure, I hope you have. If you don't, this is your warning. The leucines are not beta branched. They have a branching at the, and I guess I should have made that pink for symmetry or something. Ah, that's better. The leucines are not beta branched. And notice that the leucines are only at the D position. The valines are only at the A position. Somebody, 30 years ago in the late 80s, early 90s asked the question, I wonder if it makes a difference to have valines at A or beta branched amino acids at the A position versus non-beta branched amino acids at the D position. So they switched them. And they created this sequence GCN4PLI in which we changed the D positioned leucines to their isomer, isoleucine. Now the D positions in this new sequence are beta branched. This is isoleucine, the alpha carbon, the beta carbon, and you see that now isoleucine is just like leucine, only it's beta branched instead of non-beta branched. And then notice in this revised sequence called GCN4PLI, the A positions are leucines now, non-beta branched. So we've switched. You used to have beta branched at A, now we have beta branched at D. Used to have non-beta branched at D, now we have non-beta branched at A. That small change caused the peptide to form a tetramer. So that's a dramatic example. Well, okay, you're not gasping, so apparently it's not that dramatic. It's a striking example of the relationship between sequence and structure and ultimately function. A modest change, moving a methyl group essentially from the beta carbon to the gamma carbon or vice versa, is enough to change the structure of a peptide from a dimer to a tetramer. And that can have some consequences. For nature's purposes, the naturally evolved protein was a dimer, and it needed to be a dimer because that's the dimerization domain of a transcription factor. On the other end of these helices in this larger protein, these experiments were done on a 33 residue segment of a peptide clipped out of a larger sequence. In the larger sequence, there's a DNA binding domain over here. So the tetramer wouldn't be useful biologically, but people have learned interesting relationships, interesting rules about sequence, structure, and function from these kinds of experiments. So if anybody ever tells you that protein folding is unintelligible, that there's no rhyme or reason, you can't predict it, that's a bunch of hogwash. You just have to learn the rules. Now we have tools of AI that are helping us learn the rules, and if you take a structural biology class, you'll probably talk about Google's DeepMind AlphaFold project, which is really interesting. We have too much to do to delve more into that. The last thing I want to do is just talk about one more relationship between sequence, structure, and function, and that is in these so-called antimicrobial host defense peptides. If I were really prepared, I would have the sequence of the peptides that we have in our bodies that do this. Instead, what I have is a sequence from frogs. So I don't know why. This is called, the peptide sequence I'm going to show you is called meganin. It is from frogs. It's secreted from skin. Meganin is named after a Hebrew word for shield. So this is a defense peptide. Frogs use it to prevent bacterial infection. If you look at the sequence, I've highlighted in green all the nonpolar amino acids. I've mapped this sequence onto a helical wheel diagram. Helices that are not involved in coils are slightly wider than helices that are. So don't worry about the fact that this helical wheel diagram looks different from what we did earlier. Also, don't worry about what the colors mean or even the letters. Just understand that the green ones are the ones that are non-polar. And you can see that Maganin, like GCN4, has sort of a non-polar surface and a polar surface. Maganin does not form a coil, but what it does is it does bind to and disrupt bacterial membranes. We're going to talk about the lipid bilayer soon. That's the lecture that's posted Monday. But the lipid bilayer of a membrane has a nonpolar interior and a polar surface. And that's exactly that arrangement of a nonpolar interior and a polar surface sort of matches up with the nonpolar face of Maganin versus its polar surface. It's thought that when Maganin forms a helix, these nonpolar residues are lined up so that they can interact with the nonpolar interior of a lipid bilayer, whereas these can interact with water or the polar head groups. In Maganin, if you look at these polar head groups, a lot of them are positively charged, which is important because bacterial membranes are mostly negatively charged for reasons that we'll talk about in the chapter on lipids. So here is a cartoon of a Maganin helix with the nonpolar gray surface of the helix interacting with the nonpolar interior of the bilayer, and then the cationic or polar surface is interacting with the polar head groups. That can create holes in the membrane, and when you make holes in the bacterial membrane, the interior leaks out and the bacterial cell dies. It's actually pretty dramatic. Bacteria are packed really tightly with DNA and other things, and so when you open the membrane, they just pop, and it's like a balloon popping, and they're gone. Here's a sort of computerized image of that cartoon. You can see the Maganin helices doing essentially what they're doing here, and you can see how if you have several of these Maganin helices, they can line a hole in the membrane. And a lot of ink's been spilt on this subject. There are a lot of antimicrobial defense peptides that do this. Here at BYU, Professor Paul Savage has invented a molecule, not a peptide, that basically does the same thing, and latest research from his group has him coating the interior of endotracheal tubes with this antimicrobial compound and preventing those endotracheal tubes from being colonized by bacteria, so to prevent secondary infections in hospitals. It's really quite interesting. You should go look it up. He gave a forum here at BYU in the summer a couple of years ago where he talked about some of it. It was really fascinating. Yeah.
So when we're looking for antimicrobial disease, we should look for one that has a non-polar surface and one that has a polar surface.
That's right. That's as simple as it is. You want, for something to be antimicrobial as a helix, you want it to have a nonpolar side and a polar side. If you were to design a peptide just based on what you know now, design a sequence with nonpolar residues spaced such that one face would be nonpolar and the other face would be polar, it would probably have antimicrobial activity. The challenge is, how antimicrobial is it? And the second challenge is membrane selectivity. Will it disrupt a bacterial membrane more or less than it disrupts your membrane? Some of these peptides are highly membrane active even for eukaryotic cells, and that's bad. So there's a related peptide from honeybee venom called melatonin. It's one of the things that makes a bee sting hurt, and it will lyse your red blood cells, which you obviously don't want if you're taking a medication, right? Hello, death. So, okay. There's tons more to say, but that's all we have time for. Though, if you have any questions, I'd be happy to be derailed for even more time. So, shoot. No? Yes?
That one didn't have the.
Oh, there's two of you. Sorry. All right, let's go Emily first and then you.
How do you know if it's in an alpha helix formation?
How do you know if it's in an alpha helix or not? So, First, some of these membrane active peptides aren't in an alpha helix until they get close to the membrane. It's actually the nonpolar surface of the membrane that drives folding into a helix, so that the nonpolar side of the helix can be buried in the membrane. But we do have biophysical experiments where we can take a peptide in a test tube and determine whether it's helical or not. You will learn a little bit about that in biochemistry, but not a lot. The two techniques are nuclear magnetic resonance, which you've learned a little bit about. You can tell, for example, if you can identify this alpha carbon and this alpha carbon, there's an NMR experiment that allows you to tell that they're close to each other, so that can be a clue. There's also an experiment called circular dichroism spectropolarimetry, which you don't want to know about, and which actually I didn't want to know about when I was a student here, and now it's like the thing we do most in my lab, so go figure. But there is a tool for analyzing it. The other tool is X-ray crystallography. You crystallize your protein, you shoot X-rays at it, the diffraction pattern tells you something about the arrangement of atoms in that crystal, and you can determine whether something's a helix that way. Yeah, go ahead.
I was just going to ask, on the magnesium drug protein, it looks like it was a lot more than 3.5 to 3.6 amino acids per chain. Can it be a lot larger for a or is that just a way that they consider it?
It turns out this image, though it looks substantially different from this one, is actually not that far off. This one's oversimplified. Explaining how it's oversimplified is probably, the juice isn't worth the squeeze. You can sort of see it if we zoom in here. You don't get a linear stripe of A position residues. It sort of precesses around the helical axis, just as the two helices twist around each other. This helical wheel diagram subtracts that twist out so you can see the A and the D position surfaces. This one doesn't. And also 3.6 doesn't neatly give you 7 residues per turn, so there's that too. So this is more like if you were to actually look at a helix and just map where the residues are from the N terminus, this is what you would see. Yeah. There are helices that have, oh, I shouldn't tell you this, and it doesn't matter. The alpha helix has I to I plus 4 hydrogen bonding. There is a tighter helix that has I to I plus 3 hydrogen bonding, where coiling is tighter. That's called a 310 helix. There's also a helix that has I to I plus 5 hydrogen bonding, where coiling is looser, more residues per turn. That's called the pi helix. Neither of those are very common in proteins. Yeah?
We talked about how the hydrophobic interactions are one of the primary driving forces for
Yep.
the formation of an alpha helix. Is there a driving force for the formation of beta cleavage sheets?
Right, so what's the driving force for formation of beta sheets? Same thing, it's burial of nonpolar surface area. So what determines whether something's an alpha helix or a beta sheet? We could spend a lot of time talking about that. Individual amino acids have different preferences for the dihedral angles they want to adopt. The dihedral angles adopted by these phi and psi, the phi and psi dihedral angles for amino acids in a helix are different from those in a sheet. Some amino acids, because of their side chain structure, tend to prefer sheets versus helices. This is something that people have worked out pretty well, but it's sort of, we could talk about it, but it would just be more things to ask you questions on, I suppose, so we won't. Yeah?
What prevents the alpha helix of an antimicrobial group to make a dimer and not be able to bind?
Wow, that's an interesting question. How can an antimicrobial group peptide that has a nonpolar face, what stops that from being a dimer? And then therefore, if it were a dimer, not being able to bind the membrane because the nonpolar interior would already be sequestered. If you look at this sequence, it doesn't follow that regular pattern. There are nonpolar residues and there all are along the one surface, but it's not following the same neat 7 residue pattern, and I think that makes the difference. I don't have a better explanation than that other than it doesn't meet that sequence pattern for formation of coils. But you could do experiments that would determine whether this peptide is prone to self-associate. People do that all the time. It might be. And you would predict that the more a self-association prone it is, the maybe less likely it would be to form a membrane. That would sort of depend on some equilibrium constants, but yeah. Okay, well, we need to talk carbohydrates. So that concludes sort of peptides and proteins. Let's talk chapter 25, carbohydrates. Our goal in this chapter is I want you to understand the structures, recognize and understand the structures of key monosaccharides. What will not be new here is cyclic hemiacetals, because we've already done that. I've already actually asked you questions on exams about the open chain form of a cyclic hemiacetal that happens to be a carbohydrate. So you should be prepared to deal with that kind of thing. But we're going to talk about key structural features of monosaccharides and then more complicated glycosides, which are carbohydrate acetals. So there is going to be some facts and structure memorization here as well. I'm going to introduce you to your old friend that you maybe weren't paying attention to, but you've seen it before. And this old friend was there all along. This is a molecule that's called beta-D-glucopyranose. Most people will just call that glucose, but the chemists give it this name, and each portion of this name tells you something important about the molecule. I'm going to number the atoms, starting with the hemiacetal carbon, which is going to be this one, the one that's bonded to an oxygen and two oxygens, and one of them's an OH. This is 2, 3, 4, 5, and 6. Notice that 6 is not part of the ring. Notice that an oxygen on 5 forms the hemiacetal with the carbon number 1. We've called carbon number 1 before the hemiacetal carbon. That's fine, but carbohydrate chemists have another word for that, which you should be aware of. It is also called the anomeric carbon. I don't know why. I don't know the origin of that word. The beta part of this name tells you about stereochemistry at the anomeric carbon. If the OH is, beta tells you that the OH is up and equatorial. Or, well, I mean, if we want to be precise, we'd need to have an R versus S. But when you draw glucose in this way with carbon 6 in this equatorial position, having the OH up is the beta configuration. If the OH were down, we would call it alpha. So this is alpha D-glucopyranose. I started to hedge a little bit because, of course, you could flip the ring over and then the OH would be down, and so up and down is not absolute. When the OH has this stereochemical configuration, we call it beta. When it has this stereochemical configuration, we call it alpha. And these two, alpha and beta glucopyranose, are called diastereomers because they differ at one and only one stereocenter. They are diastereomers because they are stereoisomers but not enantiomers. And a diastereomer is a catch-all term we have for stereoisomers that are not enantiomers. They're a particular class of diastereomers. Here's a word you may not have heard before. Epimers are diastereomers that differ at one and only one stereocenter. And because they differ at the anomeric carbon, we will also call these anomers of each other. Here is the beta anomer, here is the alpha anomer. Okay, some of that's just vocabulary. Questions about alpha and beta? Okay, what's the D about? Well, the D is about the stereochemical configuration of carbon 5. And D comes from an old-timey way of talking about stereochemistry, L versus D. It used to mean, it's sort of related to left hand versus right hand, handedness. But L and D are assigned not by whether or not the molecule rotates light in the left versus the right direction, but they're assigned based on similarity, structural similarity to a parent compound, which is called glyceraldehyde. Old-timey stuff about stereochemistry that we no longer care to delve into. But when the stereochemical, when carbon 5 has that absolute stereochemistry, from our perspective in this drawing, the CH2OH is up, then it's a D amino, or sorry, it is a D carbohydrate. If everything else were the same, but the CH2OH group were at this axial down position, that would be an L carbohydrate. Most naturally occurring carbohydrates are D. There are just a few that are L. All right, so we talked about what the D is. What does the pyranose mean? Pyranose refers, well, the pyran portion tells you that this molecule is going to have a 6-membered ring structure with the oxygen being one of the atoms of the ring. That's, if you were to just say pyran, that would be like cyclohexane, only one of the CH2s is replaced with an oxygen. So the pyran of the pyranose tells you that this sugar is in a cyclic form. If someone just wrote beta D glucose, what they would mean then, technically by writing beta D glucose, and actually you couldn't write beta anymore because if it's not cyclic, there's no anomeric carbon. If someone were just to write D glucose, they would mean that your molecule is in its open chain form. So clues that you're dealing with the cyclic hemiacetal form are both the beta and the pyran. Okay? The ose tells you that it's a sugar or a carbohydrate. Things that are carbohydrates or carbohydrate based have the ose ending. So if you've heard of ribose, that's a carbohydrate. Mannose, galactose, carbohydrates. All right. The remaining portion, let's see, gluco, of course, sort of tells you what the parent compound is. That gluco name really refers to stereochemistry at carbons 2, 3, and 4. And so we will see there are other carbohydrates that if they have different stereochemistry at 2, 3, and 4, we would give them a different name here, and we'll talk about what those names are. When you draw the cyclic hemiacetal form, and the way I memorize sugars is I start with this one because everything's equatorial, and then I move on from there. In beta D-glucopyranose, everything's equatorial. Yeah.
We're talking about like stereochemistry of these, like the gluco assignments, carbons 2, 3, and 4.
Yeah.
Is it similar to carbon 1 in which it's like relative to that carbon on 5 and 6, or is it an RNS assignment? So everything seems to be relative, like based on another side chain. And then
Right, it does seem relative, and that's intentional. It is absolute, but the naming isn't necessarily systematic. So, and I'm going to have you memorize the names of 3, maybe 4 different sugars. It will be easiest to keep track of those names with these relative relationships, which is why I'm saying I would start by memorizing, because if you want to, you can assign R versus S for each of these. It's going to take you a while to make the assignments because I've done it before. There are a lot of ties, and sometimes you have to go out kind of a long way. So I don't recommend it. It's easiest just to memorize beta D-glucopyranose as being this cyclic hemiacetal form where all the OHs are equatorial and where the CH2OH, the hydroxymethyl group, is also equatorial. And then we can do changes on individual carbons. We're not going to do all of them, and we won't do all possible combinations because biology doesn't use all possible combinations a lot. All right, others? Did I miss another hand? Okay, so we've already talked about one possible change. What if the OH on carbon 1 is down axial instead of up equatorial? That's alpha D-glucopyranose. Why is it still D? Because it still has the same configuration on carbon 5. Why is it still gluco? Because it still has the same configuration on carbons 2, 3, and 4. All right? We might say that alpha D-glucopyranose is the C1 epimer of beta D-glucopyranose because it's carbon 1 that is different. Okay, so let me draw you 2 other carbohydrates that are related to beta D-glucopyranose. First is the C2 epimer. So we'll draw the 6-membered ring. We'll keep the OH in the beta position. The C2 epimer means that instead of at carbon 2 with glucose, we had equatorial down OH group. Now we're going to have an axial up OH group. Everything else is going to stay the same. Here's the hydroxymethyl group on carbon 5. This is called beta D mannopyranose. In its open chain form, which I'm not going to draw, but which you could, it would be called D-mannose. So you may have heard of mannose before. This is a sugar that's used a lot in biology. Clearly then, the beta and the D are still in place because the C1 hydroxy is still in the right place, and the hydroxymethyl group on carbon 5 is still in the right place. The only thing that's different is the OH group on carbon 2, and that's where the mannose comes from. Okay, let's take 5 and then we'll dive back into carbohydrates.